Greetings to our lecturer and audience. Today, my group and I will present for ST01402, Research Methodology and Scientific Writing for Group Assignment 2. And our theme for today is Cocoa. Introducing our group members, Chelsea Lynn Lester, Dina Hanani binti Abdul Qadir, Benedict Anak Masing, Colin Elisa Richard, Nur Faradila binti Mortedi. Our presentation will be about the instrumentations used for cocoa analysis, which consists of high-performance liquid chromatography (HPLC), gas chromatography mass spectrometry (GCMS), gel permission chromatography mass spectrometry (GPCMS), X-ray diffraction (XRD), and lastly, Fourier transform infrared spectroscopy (FTIR). The methanol was used in the mobile phase, while octadiesel is used in the stationary phase. Based on the figure, the chromatogram A is referring to the standard solutions, while chromatogram B is referring to the sample solutions. Based on the figure 1, the retention time for theobromine, theophylline, and caffeine are 5 minutes 22 seconds, 6 minutes 32 seconds, and 8 minutes 67 seconds, respectively. Based on figure 2, the data from the linear correlations between peak areas and the concentration of the theobromine, theophylline, and caffeine was plotted in a graph, and the calibration curves are linear. Based on the correlation coefficient shows that it is more than 0.9901, which indicates high accuracy, and the extractions of the components from the metal santides was complete. Differences in the retention time indicate the time of the samples adsorbed on the column packing, which is the adsorbent. So first of all, the theobromine retention time is the shortest, which means that it interacts with the column's adsorbent is shorter, and it says to be the most polar. Caffeine, on the other hand, has the longest retention time because of the longer interaction with the column packing and shows that it is less polar. The differences in the retention time for all these three compounds is affected by the polarity. Therefore, the theobromine will be eluted first, followed by the theophylline, and lastly caffeine. By comparing all the chromatograms A and B, the three components are separated completely. We can conclude that the shortest retention time indicates the most polar in the reverse phase HPLC. In terms of size, all three components is slightly similar in their molecular weight, which doesn't affect that much. From the chromatogram, we can observe that it shows a high resolution at which we can differentiate all the three peaks with different retention time and the width. Overall, the HPLC able to separate the components in the sample and identify the components by comparing with the standard solution as reference. Other than that, HPLC also provide information about the area under the peak to obtain the concentration of the compound by generating a calibration curve.
Next, we have SPME-GCMS. This instrument it was used to analyze and acquire chromatography data of chocolates which made from different geographical origins of cocoa. So when we look at figure 1, each chocolate with different geographical origin, they result to different chromatogram. And the primary sensory attribute reveal of 4 chocolate sample as nutty, fruity, floral, and spicy. Then, from this, the principal component analysis is performed to explain how this compound can relate to the primary sensory attribute. So they use the first principal component, which is 48.6%, and second principal component, which is 33.6%. The percentage of variance is explained where there is almost no variation between repeated experiments, showing that this technique gave highly reproducible result. The sample data was then plotted according to the geographical origin of cocoa. Based on the result from figure 2 and 3, the characteristic feature of the aroma compound is determined. It is revealed that chocolate sample with nutty aroma contain the larger amount of pyrazine than the other samples. And because of this compound present in the sample, it contributes to the smell of nutty cocoa and roasted aromas. In contrast, aliphatic alcohols such as phenethyl alcohols, they reside in the floral aroma type of chocolate. It contributes to the floral aromas. And this finding shows that using this technique is suitable for comparative analysis of multiple samples, and it can be used to examine correlation between aroma compound and sensory attribute of food.
Next, analysis of cocoa using FTIR. FTIR is an analytical technique used to identify and study the chemical composition of sample by measuring how it absorbs IR light. This method provides information about the molecular structure and functional group present in the sample. Figure 4 shows FTIR spectra of high molecular weight fraction isolated from raw and roasted cocoa beans of different groups. FTIR spectra of cocoa beans exhibit characteristic absorptions bands in the broad region of 400 to 4000 inverse centimeter, at 3350 indicating of OH group, and at 1650 indicating to C double bond O group, at 900 to 600 related to N-hydroglucose rings. Next, at 2622 and 2850 region correspond to CH stretching. At 1406 to 650 indicating have CO, CC, CN, CH bending, benzene ring. Last but not least, at 1020 to 1050 region suggest polysaccharides and lipid. Moving to discussion part, the port absorption band at 3350 and 1650 confirm the presence of hydroxyl and carbonyl group characteristic of various organic compounds found in the cocoa. Specific absorptions band between 900 to 600 related to N-hydroglucose ring, confirming the presence of polysaccharides. The figure print region at 1400 to 650 show distinct vibrations such as CO, CC, and CN single bond stretches, CH bending and bend due to aromatic rings. Peak in the 2922 and 2850 region correspond to CH stretching vibrations, likely from fatty acid in the cocoa melanoidin fractions. Additionally, peak in the 1020 to 1060 region suggests polysaccharides and lipid content in the sample, which is correspond to COC stretching vibrations as the bond in triglycerides and glycosidic leakage.
In this part, we will discuss the instrument used in characterization of peptides formed during fermentation of cocoa beans. GPC, or gel permeation chromatography, involves a simple solution passed through a column packed with porous gel beads. Large molecules eluted first as they are unable to enter the porous gel beads. Smaller molecules are eluted later as they can enter the pores and navigate through the structure. Smaller molecules have longer retention time as opposed to larger molecules. The molecular weight distribution is determined by measuring elution time against non-standard. Figure A shows the partial GPCMS chromatogram of cocoa extracts at different fermentation stages. It shows the changes in relative abundances of components in the region where peptide elutes. It also shows the increasing quantities of small peptides and amino acids, proving the hydrolysis of polypeptides. Meanwhile, in Figure B, it shows the GPCMS chromatogram of fermented 5 days cocoa extract. It shows the species that form ions eluted in the time window between 12 and 25 minutes. It coincides with the retention time window of eluted standard amino acids and peptides. Out of the many peaks shown here, we will discuss the two most significant ions that are located at MZ621 and 902. The maximum intensity of both ions is at retention time 24 minutes, which is later than octapeptide angiotensin II, possesses hydrophobic properties, and contains aromatic residues. Both were then analyzed using sequence to match them with sequences of vicillin and albumin. The ion in MC621 matched to a hexapeptide, and the ions in MC902 matched to a nonapeptide. This shows that during fermentation of cocoa beans, the larger peptide molecules will be hydrolyzed into smaller peptide molecules.
X-ray diffraction XRD analysis is an essential technique for characterizing the crystallographic structure, phase composition, and other structural properties of materials. For cocoa analysis, XRD can be used to study aspects like polymorphism in cocoa butter, involve examining the diffraction patterns produced when X-rays interact with crystalline structure of the sample. H polymorphic forms such as alpha, beta prime, and beta displace distinct diffraction peaks at specific angles to theta, allowing for qualitative identification of the crystal phase present in the sample. For instance, as the cooling rate decreased from 5 degrees Celsius per minute to 0.5 degrees Celsius per minute, the onset temperature for crystallization shifted to higher values, and the stable beta phase became more prominent, demonstrating that the slower cooling facilitates the formation of more stable crystal structure. The XRD results are further supported by the SC data, which provide insight into melting behavior associated with each polymorph. For example, the SC curve may show the distinct melting peaks corresponding to transition from alpha to
alpha to beta forms, confirming the stability and quality of chocolate products. The presence of specific diffraction peaks associated with form B, considered optimal for chocolate production due to its stability and desirable melting characteristic, was identified in the XRD patents. This correlation between XRD and thermal analysis is crucial for chocolate manufacturers as it helps optimize processing conditions to achieve the desired texture and shelf life of chocolate products. By monitoring changes in crystallization behavior through XRD, producer can ensure that cocoa butter maintains its optimal polymorphic state during storage and processing, preventing undesirable phenomena such as bloom on chocolate surfaces due to improper crystallization.